Okay, so thank you for this opportunity. I'm a postdoctoral fellow in Dr. Arboleda-Velasquez laboratory, and I will be talking about active mechanisms of resilience against familial Alzheimer's disease. Specifically, I will be discussing about two of our recent publications. First, I will cover some of the major findings of the APOE Christchurch homozygote case. And then I will discuss our recently discovery on the resilience to autosomal dominant Alzheimer's disease in a really bulbous heterozygous man. Just to give a little bit of a background from where we come from with our studies. So this is a long project and a highly collaborative multi-PI project that is focusing for years on characterizing the world's largest known kindred with autosomal dominant Alzheimer's disease. We are currently characterizing more than 1,200 of these kindred that are characterized by this autosomal dominant mutation in the presenting one gene, referred as E288 mutation, that leads to a median age of onset of dementia by 49 years, with a competency interval of 5% between 49 and 50 years of age. So it's a very aggressive form of Alzheimer. And these presenting-1 carriers show faster rates of amyloid beta deposition since presenting-1 is part of the beta secretase complex that along with the base 1, so the beta secretase complex and the gamma together, they're involved in the processing of the amyloid precursor protein that leads to the formation of amyloid beta. And also we found this rapid tau accumulation, which is the second hallmark of Alzheimer's disease. What we discovered in 2019 was a female individual with confirmed autosomal dominant Alzheimer's disease that did not develop mild cognitive impairment until her late 70s. So we almost have 3 decades of delay of the age of onset of dementia. So using brain imaging, so what I'm showing here are PET scans on the top for the Pittsburgh compound to detect the amyloid burden, and on the top tau PET to detect the tau burden. We confirmed by comparing the protected case with the homozygosity for APOE3 Christchurch to MCI carriers at typical age with the same mutation. We have a higher amyloid burden compared to the MCI carriers. You can see and the bottom, we can see risk to the other MCI cases. We also quantified the cerebral metabolic rate for glucose, as we can see here with these counts. And when we compare this case to the MCI carriers, we can see the preserved glucose metabolism as shown by this higher intensity in red and highlighted with the white circle. So therefore, we observe limited tau pathology and preserved glucose metabolism despite increased amyloid pathology. Here is just a comparison, more quantitative comparison of the hallmarks that I just described earlier. The amyloid burden, we can see older MCI carriers, the APOE Christchurch case has a high amyloid burden, little reduced compared to the younger MCI carriers. Of course, similar to the AMPER carriers toward the higher end, because these are individuals with the presenting-1 mutation, even though it's protected. And we have also characterized the hippocampal volume, the cluster with the younger MCI carriers, and the preserved glucose metabolism, as we can see, the red dot that is referred to the Christchurch compared to the gray that are the AMPER carriers. So just to go more deeper in understanding what mechanism of protection is helping this delay in this patient of cognitive decline, just I wanted to give an overview of the APOE variants. So from the chromosome 19, APOE actually is known to have several variants, APOE3, APOE2, APOE4, and the newly discovered protective variant, the Christchurch, the one we discovered along with Jacksonville, recently discovered along many others. What I want to point out, while APOE3 is physiologically neutral, APOE2 has been associated for lower risk for Alzheimer, along with our protective variants I just mentioned, whereas APOE4 has a higher risk for Alzheimer, specifically sporadic Alzheimer's disease. This is an important concept because even though there is just one amino acid different that differentiates these variants, this is a very important in terms of how the structure is affected of APOE and how this reflects in terms of the function. Function that is heparin binding, lipid binding region, and this can be due to the different secondary structure that due to this one single mutation the APOE variants can have, as well as post-translational modification that can be affected due to the differences even if it's just one amino acid. So to go also on reviewing the biochemistry, APOE is physiologically secreted by astrocytes, it's lipidated and interacts, which has been known from the literature, with glycosaminoglycans. And that helps for the binding to the apolipoprotein receptors. I'm just summarizing two of the relevant in the neurons, LDLR and LT1. And this is important for the downstream mechanism of APOE in the lipid transport, amyloid beta clearance, neurogenesis, synaptic repair. What we want to understand with our studies is what is the effect of the Christchurch mutation in the binding of the glycosaminoglycans, which was still fully elucidated. So we hypothesized the protective mechanisms against Alzheimer's disease-related neurodegeneration depend upon the efficient interactions between APOE and glycosaminoglycans, or GAGs. To compare the APOE Christchurch to the other APOE variants, we modeled the glycosaminoglycans in a cell-free system using heparin affinity chromatography that allow us to study how APOE can interact with heparin using high salt concentration to destroy these polar interactions between APOE and heparin. And what I'm showing here is an anti-histag Western blotting of this recombinant APOE variants that we use to test the interaction with heparin. And I'm having here on the top APOE 4, followed by APOE 3, APOE 3 Christchurch. And this Western blotting shows where from the heparin concentrations of sodium chloride. And we can see APOE 4 has the strongest affinity, and therefore we needed a higher salt concentration to elute APOE 4 compared to APOE 3, whereas the protective variants, APOE 2 and APOE 3 Christchurch, were observed to have the reduced affinity to heparin since we used less salt to destroy these polar interactions. We confirmed this also independently using ELISA. This is the anti-histag ELISA, where we can see the profile of the eluted fractions, and we can compare in black APOE 4 that has the highest affinity compared to APOE 3, and again APOE 2 and APOE 3 Christchurch having the most reduced affinity with heparin. Specifically, we observed APOE 3 Christchurch having an even lower affinity for heparin since it was a very little salt needed to elute APOE 3 Christchurch. So we also tried to see, since it's been known from the literature, the glycosaminoglycans and APOE have a role in amyloid aggregation. We also tried to see whether there was any effect of APOE 3 and APOE 3 Christchurch in changing the amyloid aggregation pathway. And what we use is a Q-flavin T assay. This is spectroscopic assay. They use the indirect ability to Q-flavin T to fluoresce when intercalates in the formation of beta sheet structure that are typical of the amyloids. So we can see here each profile is it reflects aggregation status of amyloid beta. And what I'm showing here with this diagram is an explanation of the phenomenon that we can observe with Q-flavin T. So amyloid beta first is a monomer misfolds, and we have this initial nucleation phase that allow the formation of the seeds that are small oligomers that are packing in the formation of protofibrils and mature fibrils. When the beta sheets is mostly abundant is where we have the highest intensity of emission of the Q-flavin T. So we can use this as a quick assay to determine the aggregation kinetic of amyloid beta. What we observed was that APOE 3 Christchurch increased, as we can see with the red, the round dots increased the amyloid aggregation in a significantly higher way. This increase was still higher than the native amyloid beta, but reduced compared to APOE3 wild type in the presence of the Christchurch mutation. We then compared using a luciferase assay in vitro the effect of the two variants, and we confirmed that APOE3 Christchurch has a less effective is a less effective trigger of amyloid beta aggregation as compared to APOE3 wild type. So reduced amyloid beta aggregation in the presence of the R136 mutation is the conclusion that we had for this study. So the next step, and this opened our new field that is now ongoing, was to try an approach to mimic this loss of interaction between APOE3, Christchurch, and heparin for translational purposes. So in this study, we presented an antibody referred as A1334 as a novel negative modulator of APOE heparin binding. So we tested the hypothesis that to design an antibody that targets the Christchurch binding site, we can reduce the heparin binding affinity to model the possible loss of APOE to glycosaminoglycans interaction in vivo. So what I'm showing here on the left is an anti-histone Western blotting similarly to what I showed before, and compared APOE3 with control and APOE with the antibody, the control being the buffer in where the antibody was prepared. And we can see here in the ELISA with a similar approach that the area under the curve that we can see in magenta of APOE3 only exposed to the control was significantly reduced in the presence of the antibody A1343. Thus suggesting that the A1343 antibody anti-APOE HSPG or glycosaminoglycans binding domain successfully reduces the heparin binding affinity to APOE3. So it can be used as a candidate for future drug development. So in summary, for this publication, we discovered the first protected case of familial Alzheimer's disease. We confirmed that the protected case was homozygous for the APOE3 Christchurch variant. We hypothesized that the protective mechanism was linked to the loss of binding of APOE to heparan-sulfate protoglycans in the presence of the Christchurch mutation. The protective APOE Christchurch variant may have a role in modulating amyloid beta aggregation, and then to design anti-APOE Christchurch antibodies may be a successful strategy to mimic the loss of APOE heparan-sulfate protoglycans. So now the focus will be on the next discovered protected case that here is referred as the RELN-Colvos case. So we also use the same approach of analyzing both amyloid and tau PET scans of this RELN-Colvos case that was presented with the presenil-1 E288 mutation, so it was part of the same families with this autosomal dominant form of Alzheimer's disease, and we compared this case to the presenil E288 mutation carriers with MCI similar age. And I'm showing here on the top the amyloid scan with Pittsburgh compound, having a similar trend of amyloid burden compared to the MCI carriers. But on the bottom, when we compare the tau burden, we observe the reduced tau burden when we compare this protected case to the MCI typical age carriers, similar to a profile that we observed for the first case, the Christchurch mutation homozygous. Similarly to the first case, the cerebral metabolic rate for glucose was preserved in the same parietal occipital region as compared to the MCI onset of typical age carriers with the presenil-1 mutation. So preserved cerebral metabolic rate for glucose and reduced tau burden in the RELN H3447R variant or RELN-Colvos despite the increased amyloid burden. So we also analyzed post-mortem the brain of this case, and what I'm showing here on the left is the CA1 region of the hippocampus and the entorhinal cortex. So we confirmed the presence of both amyloid beta and phospho-tau as hallmarks for the Alzheimer's disease pathology in this protected case. And we also compared the entorhinal cortex regions of the RELN-Colvos to the APOE Christchurch for amyloid beta and tau, and compared them to familial Alzheimer's disease and sporadic Alzheimer's disease. As we can observe here, we have certain areas, specifically in this entorhinal cortex region, where we can see reduced phosphatol levels along where we observe the same in the APOE Christchurch, and not much reduction in the amyloid accumulation in the RELN case compared to the familial Alzheimer disease case. Actually, we can even see higher amyloid in these regions in the RELN case compared to the familial Alzheimer disease. So we then use and compare the RELN case to APOE Christchurch, familial and sporadic. What we observe is that the higher neuronal density in the clinical case and higher neuronal density in the COA region of the APOE Christchurch case compared to the familial and sporadic Alzheimer's disease case. Here, what I'm showing is the hippocampal and parahippocampal regions of the RELN case stained with the Kluver barriera, and also compare the levels of RELN in the RELN case to the APOE Christchurch case, familial and sporadic Alzheimer's disease case. What we can see here in this staining is that the higher Kluver barriera staining, so higher neuronal density, and also higher positive staining to RELN in the RELN case. So just to go a little bit more into the detail was where this mutation of this protective case is located. I'm just showing here a diagram of the RELN protein. RELN is a very large protein, more than 480 kilodalton, and it's been known to have three major domains: an amino terminal domain that is important for the dimerization and the assembly of RELN, an intermediate domain that is known to be important for the receptor binding domain, and the C-terminal region that is being proposed to be important for the secretion of the protein, and also has been known to have a role in the binding to interacting partners, such as European-1. But overall, so far, this C-terminal region of RELN was poorly studied. So we wanted to fill this gap of knowledge as well. The mutation of the protective case is specifically located in this CTR region of the C-terminal region of RELN. And here is in gold shown where the H347R mutation, so the substitution in R gene, is located as compared to the wild type. Just to review the biochemistry of RELN, and this brings, in my opinion, the two cases together. RELN biologically is known to interact with the apolipoprotein receptor. And here I'm showing this diagram with LDLR as an example. And what is known, RELN actually interacts as an agonist. APOE is known to be an antagonist for RELN. So they are involved in a similar biochemical pathway that is extremely important for the downstream protective non-effect of RELN in reducing the hyperphosphorylation tau through phosphorylation of the AB1, disinhibition of GSK3 beta are the latest chain link that is involved in the protection against the hyperphosphorylation of tau that leads to neurofibrillary tangles and platform and neurofibrillary tangles formation. So what has been shown earlier is APOE is known to have a critical binding and loss of binding in the case of the protective variant APOE Christchurch. And this is also a step that is known to be relevant for the biochemical pathway that is downstream of APOE. So what we wanted to know now, what is the relationship and the protein-protein interaction that is involved in RELN overall, so RELN wild type to fill up this gap of knowledge, and whether the mutation will have any effect in changing this specific protein-protein interaction. So we hypothesized the H347R mutation leads to a gain of function due to increased binding to glucosaminoglycans, thus resulting in increased level of phospho-DAB and reduced tau hyperphosphorylation. We used a very complex approach because, as I mentioned earlier, our goal was to fully characterize the poorly understood C-terminal region of RELN. So we did a functional characterization of the C-terminal region for RELN using heparin affinity chromatography and binding assays. We also characterized the structure of the C-terminal region for RELN wild type, and also did in vitro and in vivo characterization of the H347R variant. What I'm showing here in this little busy slide is the analysis using chromatography, heparin affinity chromatography specifically, of a small library of reading variants, including the protected mutation, the APOE, to lysine aspartic acid as compared to the wild type into forms. A form that is being referred as long. So there is the full 36 amino acid, and in the bottom there is the full list of the amino acid sequence of the small library that we use to analyze. And the variants expressed as short, that are lacking of the last 6 amino acids, since we are aware that in physiological condition, the last 6 amino acids are actually cleaved by furin. So we wanted to see also in a more close to physiological conditions whether there were any differences in heparin binding using heparin binding chromatography to model again the interaction between reading and glycosaminoglycans in a cell-free system. In blue are the chromatograms of reading wild type long and short, and in green the R variant compared to the other 2 variants used as control. We can observe that actually there is an increased binding because there is a higher salt concentration and elution of later retention time in the presence of the arginine mutation, thus suggesting that the reelin in the presence of this colbos mutation binds with a higher affinity for heparin as compared to the wild type or the other variant. We tested using the surface plasma resonance the binding to determine the affinity constants between the heparin and the both reelin wild type and reelin H247R C-terminus region. And what we observed actually under these conditions, there was not much differences in terms of the KD. But what I would like to bring to the attention actually, when we analyze the response, the first part of the graph is the K on, so the interaction, and the K off, the release, actually has higher K on, slower K off. So that might be one of the possible effects, even if it's very small, of this mutation compared to the wild type. So then, to resolve the structure of this C-terminus region, which was not known before, we use the first circular dicrotism, this is the graph on the left, and to determine whether this C-terminal region had a secondary structure. And what I'm showing here in black conditions, really very difficult under native conditions. However, in the presence of 3-fluoroethanol, we were able to force an alpha helix secondary structure that allowed us to resolve using 2D NMR the C-terminus region structure. Here is an in silico modeling of the structure that we were able to obtain upon 2D NMR analysis. And this is a video that shows more in detail how this C-terminal region is so flexible and dynamic. And that's why most likely so unstructured, because actually it's a highly flexible, even though in the first part of this C-terminal region, we can see a more stable part, less flexible, and then a highly flexible and C-terminus region. So this allows us to hypothesize that we have what we call the flexibility vertex domain, from where the rest of the amino acids have different orientations. And here I'm showing in magenta the arginines that are oriented on the same side versus in cyan, the other oriented on the opposite side. This arginines is the one that we believe are the most involved in the interaction with the heparan-sulfate proteoglycans or glycosaminoglycans. And we hypothesize the presence of a first alpha GAG binding site that is physiologically cleaved by furin. And therefore, what is important, and maybe the difference stays in what the mutation does into the beta GAG binding site by having this extra arginine that supports this hypothesis of the increased polar interactions that allow this increased glycosaminoglycans in stronger interaction. So we also wanted to confirm whether this reading mutation was having any effect on the apolipoprotein receptors. And what I'm showing here is the VLDL-R receptor binding the ELISA of renin intermediate domain that is used as a standard to renin wild type, renin H344R, and a combination to mimic what we have in the heterozygous protected case. And on the bottom, we also tested the C-terminus region alone to test and confirm whether there was any binding on this region to the apolipoprotein receptor, specifically VLDL-R on the left, APOE receptor 2 on the right. And we did not observe any differences in binding between the two variants for the apolipoprotein receptors, nor any binding of the C-terminus regions alone. Therefore, this supports the hypothesis that these differences and increased effect that we observed from the RELN-Colbos variant is specifically for the glucosaminoglycan binding. So then our next step was to test the downstream effect of whether we could see any changes in the presence of the RELN-Colbos mutation in the downstream signaling of renin. So we used mouse cortical neurons and treated either with MOC or wild-type renin or renin H344HR, and we tested the changes in phospho-DAB, as I mentioned before, is the downstream effect of the renin interaction to the apolipoprotein receptor. And we observed a significant increase in the levels of phospho-DAB when we treated the cells with the Colbos variant or H344HR, since we generated the murine homolog that is in position H344HR that is equivalent to the human variant H34447R. So then we wanted to have a more complex system to test whether in vivo we were able to observe the same gain of function that we hypothesized with the findings I just showed with cell-free and in vitro system. So we designed a knock-in mouse model referred as a mouse renin H344HR or mouse Colbos, and we used amplification of genomic fragments containing homology arms using fidelity taq DNA polymerase. And then we did the assembly of these homology arms into targeting vector together with the recombination site. What I'm showing here is the wild type renin was substituted to generate a knock-in. And as I mentioned, the H344HR is homologous to human H34447R renin. So we did a neuronal characterization using crescit violet and Kluver-Barrera staining of the cortex, the biomolecular characterization of renin, and we did the renin levels, DAB levels, GSK3 beta levels, and also we tested the immunohistochemically tau levels, and also we did the behavioral analysis as well. So what we first wanted to confirm whether this renin variant was affecting the gross anatomy of the brains of either heterozygous or homozygous renin brains, because it's known that a loss of function of renin actually leads to impaired synaptic formation, synaptic plasticity, and what are referred in the literature as the Wheeler mouse. They lose the structure of the cerebellum. So what we can see here on the top is the three independent mice slides compared to the heterozygous and the homozygous renin mice. And we did not observe any gross anatomical changes, nor in the cerebellum when we compare the brains. So then we wanted to see more deep, and we focused our attention on the cerebellum, since in the literature there are extensive studies of the role of renin in the cerebellum. So what we observed when we analyzed the granular the renin was type to heterozygous and homozygous renin mouse compared to the wild type. So we concluded that there was increase in neural density in the granular layer of the cerebellum of the H344 renin homozygous mouse. And this was similar to what I showed earlier in the human cases. We observed increased neuronal densities as well. So we also tested the renin levels, and what I'm showing here are representative Western blottings of female and males. And here on the bottom is just a cartoon showing that actually RELN was processed. So we have around 400 kilodalton, what is detected as RELN full length. And then all the other different fragments, NR6, NR2, R38, R36, R78. And as I mentioned before, the amino terminal region of the RELN is important for the oligomerization. So anything that was higher than this molecular weight, we refer as oligomers for RELN. And we compare the females and males age between 6 and 12 months of age. And when we quantified all of the different fragments, we actually observed a significant reduction of the full length levels of RELN in the males, no significant changes in the females. Same in terms of the RELN R38 fragments was significantly reduced in the males, none in the females. No changes for the fragment R78. So we concluded the levels of full-length RELN detected by this anti-C-terminal RELN antibody were significantly reduced only in the RELN H3448 male mice. Here is using a different antibody. It's called the CR50 antibody to target also the amino terminal region. And we observed a similar pattern. When we analyzed the positive bands detected by this antibody anti-amino terminus RELN, we did not observe any significant changes for the female mice, but significant reduction in the levels of this fragment detected by this anti-RELN antibody only in the males. So again, levels of this NR2 RELN fragment detected by the anti-amino terminal RELN antibody are significantly reduced only in the RELN H3448R male mice. So we then tested levels in phospho-DAB, and what I'm showing here again, the comparison between females and males. And in the same pattern, we observed significant changes in terms of increased levels of the phospho-DAB levels in the males compared to the wild type, but no significant changes in the females. So similar changes for the mice that were carrying the RELN polvo, so the RELN H3448R variant. We tested the levels of the phospho-GSK tyrosine 216, serine 9, total GSK, and quantified these levels in both females and males. And we observed a significant increase in the male levels for GSK tribeta serine 9, no changes in the females, and no changes in the tyrosine 216 GSK tribeta for males and females. So then our next focus was to determine whether this mutation of the RELN gene was having any effect on tau, to give also a pathological readout to see whether it was protective under a pathological background. So we used the tau P301L transgenic tau mice and crossed these mice to the RELN mutated mice and test whether this protective effect in terms of increased phospho-DAB and possibly if there was any reduction in the hyperphosphorylation of tau was observed in this cross mouse. The P301L mouse presents usually before 6 months of age tau tangles, and after 10 months of age, neural loss and gliosis. Behaviorally, there is this known phenotype that can be observed and assessed and scored of this back post hinge reflex when you elevate the tail. So we can determine and score the response to tail elevation of this mouse and determine whether these mice present any motor impairment. So this is what we did. What I'm showing here is just a representative observation that allow us to score the mice as severely impaired, score 0, halfway impaired, score 1, or physiologically normal, score 2. And we compared the transgenic tau mice to the mice that were crossed also with RELN H3448R, and we observed a significant rescue of the phenotype in the presence of the RELN mutation. So then we also wanted to see whether the RELN mutation was having any effect in reducing phosphorylated tau or neurofibrillary tangles. So what I'm showing here is a representative staining of the hippocampus, comparing wild type, RELN homozygous with the colobus mutation, compared to tau, or the cross tau to RELN colobus mouse variant. And we observed a significant reduction of the neurofibrillary tangles and neuropil threads in the pyramidal neurons of both CA1 and gyrus as compared to the H-tau transgenic mice. So in summary, we discovered the second protected case against autosomal dominant Alzheimer's disease. Post-mortem analysis of the RELN colobus case revealed the role of the H347R mutation in increasing neural intensity. We characterized the effect of the H347R mutation in vivo and in vitro, and determined that the gain of function of its function is also to increase binding to unsolved proteoglycans. Biomolecular analysis showed that the genotype effect is sexually dimorphic as affects the males and not female mice, as I showed with the Western blotting analysis. The presence of the mouse RELN colobus variant leads to differences in RELN levels and increased phospho-DAB levels and improved behavioral performance. So we hypothesized that the RELN colobus is a promoter of resilience against autosomal dominant Alzheimer's disease. With that, I would like to thank my supervisor, Dr. Arboleda-Velasquez, the lab, and the multi-PI project, so the Mass General Hospital, the Gruppo de Neurosciences de Universidad de Antioquia, Dr. Glofera, the Harvard Aging Brain Study, the Institute of Neuropathology, supports, but most importantly, the Colombian families. And thank you for listening.